Everyone's aware. Great. Okay, and if there are any issues with viewing my screen or hearing me, as I said, please let me know via the chat so I can take care of that. All right, so I'm sure that you all know a bit about IDseq since you have made it to this webinar, but I just wanted to go over a little bit about what IDseq is and a little bit of its background. So IDseq is an open source cloud-based service for pathogen detection and surveillance. It is a tool that was developed at CZI, or the Chan Zuckerberg Initiative, which is a philanthropic technology company, but it was developed really in close partnership with the Chan Zuckerberg BioHub, which is a nonprofit group of scientists, and we still work really closely with that group in order to make sure that we are developing a tool that is solving real needs of scientists. The three main use cases for IDseq can be summarized as discover, detect, and decipher. So discover really has to do with identifying the pathogen landscape across a cohort of samples. Detect is more focused around monitoring and reviewing potential outbreaks. This is more on the surveillance side in a particular region or worldwide. And then decipher is this medical mystery case that we'll see later on within the tool, actually, that is used to find potential infecting organisms in these large data sets. A couple of things that distinguish IDseq from similar tools are fresh data sources. And by this, I mean that we update our index database that we pull from NCBI fairly frequently because we want to allow users to find or really compare against the most recent database and make sure that we're delivering value there. We are open source, so you can find our code on GitHub, and I definitely encourage you to do that and poke around there if you're interested on that side of the tool. We are committed to always providing IDseq as a free service, so we take on that computational cost. You can access IDseq anywhere online, and so it's really easy to jump on the tool and start your work no matter where you are, as long as you have an internet connection, pretty much. And lastly, I just wanted to highlight that IDseq is safe and secure, and that we take privacy very seriously on our team and make sure that we are following best practices in that area. And then finally, before we actually jump into the tool itself, I wanted to show this slide as a way of highlighting that IDseq is part of this larger integrated workflow. And for those of you that are more familiar with metagenomics or how work like this runs, this might look like a very familiar diagram, but I'll just run through it really quickly for those that aren't. IDseq is represented in this drawing by the dotted box. But if we actually start over here, this process begins by gathering samples. These can be human samples, they could be animal samples or insect samples. Once you have a sample like blood or CSF, you can actually take that sample, bring it to a lab, and that's where the actual sequencing will begin. There's several library prep stages. We call this the wet lab portion of this entire workflow. When the samples have actually run through the sequencer itself, it typically outputs these data files in the format of FASTA or FASTQ files. And this is actually what we end up, or what you would end up uploading to IDseq. And this is where we really come in. So once you upload those data files to IDseq, that will automatically kick off this pipeline, our pipeline within IDseq, that starts isolating host and non-host reads. And this is in an attempt to identify what microbes or other organisms are present in the sample. So as I said, this is this categorization that we're doing. And for IDseq, we are really interested in those non-host reads. So we actually will throw away those host reads to surface what are the potential infecting agents there, or what is the landscape of microbes within this sample. This is particularly important just from a human, especially from human hosts, because we want to make sure that we actually, don't want to hold on to those human reads from a privacy perspective. And that's really not the aim of the tool either. It's to bring to light what those non-host reads are. And then the final step in the pipeline is this analysis. We provide several bioinformatics analysis tools that can happen through our user interface, and I'll go over a couple of those when we jump into the tool. Okay. The last thing that I wanted to say before actually doing that and starting the demo is that just highlighting again that IDseq is a tool intended for research or academic use, and it's not intended, and actually our terms don't allow you to take the data and use it for monetary gains. So just highlighting that here as something to be aware of with this tool. All right, now I'm going to jump into IDseq itself. So when you log into IDseq by just going to idseq.net, you're taken to this home screen. What I'm looking at here are any of the projects that I have been involved in personally, either by starting the project myself or by uploading samples there. And you can tell that where I am in the tool by looking at this My Data tab that's highlighted right now. When you first enter the tool, you will most likely not see anything on this page because you haven't uploaded anything to IDseq yet. So what will probably be most interesting for you is to head over into this public tab. So what we're looking at now are all of the projects in IDseq that are public. And by public, I just mean accessible to other users in IDseq. Because we are still in this closed beta, that means that these projects and samples are not accessible right now to folks without an account. So you'll see for each of these projects, the name, a little bit of information about it, when it was created, and some metadata. It's a good way to start exploring these projects and understanding a little bit more of what's out there. I'd say another great way to do that is by looking in this right-hand panel that has metadata about all of the projects in this grouping, and you can explore that a little bit more and start digging in that way. Finally, you can go over into the left-hand panel and actually start filtering this data by these different categories. So for example, if we only wanted to look at projects that had mosquito hosts, that's pretty easy to do just by applying that filter and then starting to explore from there. I'm going to switch over into the samples tab now, which is just a different way, a disaggregated way of looking at this data. Now we just see a flattened list of all of the samples. So you see the sample name, the uploader name, the project that it's a part of, and then some metadata about those samples. Some of these, as we can see, are actually quality control metrics, and you can use these to start to assess the quality of your library prep and how the samples pass through our pipeline. And I would encourage everyone to go and read more on our Help Center to understand a bit more what these metrics are and how you can use them in your analysis. All right, and then from here, one of the things that I'd like to show is that if you are interested in a particular taxon, one way to find the samples that have that is that you can actually go up here and just start searching for it. So if I'm looking specifically for samples with pneumonia, I can just type that in, select that taxon. You'll see that actually applies the filter right there. And then I am left with the samples where that taxon was found. I'm going to remove that for now. Another really cool way to start to visualize what is in IDseq is actually in the map view. So if I click on this little globe icon here, I can now look at a global map that shows the distribution of our samples across the world. And these are showing where those samples were collected. So if I want to dig further into these samples that were found in Germany, I can click on them, find out some more information at a high level. I could then dig into the samples themselves and understand a little bit more about what was found in those samples and what part of the study they were. And right now we're just looking at, again, the samples that have become public and those that have this location data attached to them. All right, I will switch back to this list view here. And now I'm going to jump into a particular project. So if I go back to my data, I'm a part of a project called Medical Detectives. So if I click on this, I am taken to this page, this project page that is an encapsulation of about, I think about 15 samples. And this is actually this particular group, this project is a group of real cases from around the world where the researcher didn't know what was making the patient sick. And so they wanted to use IDseq to see if metagenomics could help surface what that infecting agent was in these cases. So let's look at one patient in particular. Scroll down to patient number 9. This was a cerebral spinal fluid sample. If I click on that sample, I'm now taken to the report. I already have some filters applied to the report page here. As you'll see, metagenomics is extremely sensitive. So I started off with around 6,600 total rows showing the taxon that were found, or the taxa that were found. But with these filters applied, I'm able to start to narrow it down based on I'm interested in viruses with these particular characteristics. And so what's really interesting about this case is that this was a young child in Bangladesh with a fever of unknown origin and clinically suspected meningitis. And this case was particularly challenging because almost all of the normal suite of tests that this person was given came back negative. But actually with metagenomics and using IDseq, they were able to discover that a potential infecting agent was this chikungunya virus. You can see here, there's a bunch of metrics that were surfacing, but this RPM, or reads per million, is a relative abundance metric, and it's a good place to start when looking and seeing what are potential infecting pathogens. And you'll see that these numbers are far and away greater than the other microbes or the other viruses that meet these conditions. And so this was a pretty clear-cut case of, we think this is the culprit. Not everything is this cut and dry, but I think by learning a bit more about how to use these metrics and interpret the data for your samples that comes through in this report, you can really start to dig in and understand what's present. This is pretty cool too, because this discovery actually ended up affecting public health decisions within Bangladesh where this sample was taken based on these results. So now I'm going to switch from this view of a report of a single sample into one of our other bioinformatics tools, which is a heat map. So you can create a heat map in IDseq by selecting any number of samples, and it will kick off the process of generating the heat map automatically. This one I had already made myself for this demo by applying, again, a set of filters based on what I knew I was most interested in here. You can adjust these, adjust the view settings to create a heat map that makes the most sense for your research purposes. In this case, this was actually a group of cats, cat samples, that the researcher was interested in looking at and discovering what was making these cats sick. So if I'm going to collapse the top section here around filters and these view settings so we can just get a larger view of the heat map. But if we take a closer look here, and I'll actually zoom in on the heat map, we can see that the creator of this heat map added this metadata, diseases and conditions. This was a particular piece of metadata from their study. You can add any piece of metadata that you'd like to the heat map as long as you uploaded it in with your samples. And these are really a great way to start to begin to attribute patterns that you're seeing in your data, start matching them up with these metadata that you upload. So if we look here and we actually sort by this metadata, I'll zoom out again a little bit. One of the things that you can see is that here are our sick cats marked by this purple. That's the water control in red, but then and in this brown. And one of the things that you can notice and that the researcher found was that these cats, the sick ones, all had prevalence of this feline calcavirus. And so that to them was a great discovery that they could use to start investigating what this feline calcavirus was and why it was appeared to be the infecting agent in so many of this cohort of samples. And if we click on this, we can learn a little bit more about it here and follow any of these links to understand a little bit more if you see a virus or bacteria that come up that you're unfamiliar with. All right. And then the last thing that I'm going to show really quickly is our upload flow. So if I leave our heat map and go over to that upload tab, if you do have samples that you're ready to upload to IDseq, excuse me, you can do so here. You can also do so in the command line. You can create a project, but I'll just use one that I have already created just for this test. And then I can either upload the files through my computer, but I can also upload through BaseSpace. We do have an integration there, so you don't have to download the raw files, which can tend to be really big to your local machine. So here I have two FASTQ files, so I'll grab those and upload them. Press continue. And now I can upload metadata about these samples. I can either do so right here by selecting the host organism, what type of sample it is, if it's a water control or not, what type of nucleotype, the date that I selected or that I collected these. Oops. And let's put in a location. Great. If that looks good, I will click on metadata or click on continue. Now I'm taken to this final review step, and really here, I'm just looking through what I have already input. And if this all looks good to me, I also will review this box down here that tells me what host is being subtracted. Here, because I've chosen the mouse as my host organism, and we have the mouse genome in IDseq, we will subtract out the mouse genome as well during our host subtraction step in the pipeline. The last step is just to agree that to our terms and conditions, and then kick off the upload. I'm not going to do it right now, but that would kick off uploading to our pipeline, and that starts processing right away. And you can actually track in IDseq as those samples start to process. Great. And then since we have a couple minutes, I did want to show one other thing pretty quickly. Let's go back to that medical detectives project. And if I look at that patient 9 again, one thing that I did want to show is that we are able, you can look at this either in this table view, you can also view it in a taxonomic tree view and explore the different nodes in this way, if that's of interest. And I also wanted to show that by clicking on our pipeline version that the sample was run on in the upper left-hand corner, we're actually taken to a visualization of our pipeline. And this is a great way to understand and dive in a little bit more deeply to what's going on in the pipeline. And it's also a great learning tool if you're unfamiliar with metagenomics or how these pipelines run. So you can expand each step and dig a little bit deeper, learn what actually happened in that step, what the input and output files are. If we look deeper, a lot of times what you'll see is even the steps themselves and the commands written out completely here so that we can provide more transparency into how our pipeline is running. If you do have any questions beyond this, I would recommend definitely checking out our Help Center. So I can do that just by clicking on my name in the upper right-hand corner and clicking on Help Center. You can also just go to help.idseq.net. And this has a lot of these user guides, which actually will walk you through analyzing a single sample, analyzing multiple samples. It's a great place to start, especially if you're unfamiliar with some of these tools. And within these sections, you'll find much deeper explanations of the metrics on these pages, and as well as some other information to help you with the interpretation and analysis. And then there's some pretty helpful FAQs in here as well. So definitely would recommend that you check this out or head over here if you do have questions. All right, I will pause here for a second and give some time for any questions. So again, if you do have questions, please feel free to put them in the chat or in directly in the Q&A. Okay, I see one question that came in. On the table for chikungunya virus, did one of the metrics include % coverage of the genome? Okay, great. So let me head back there. I can actually show you something specific to that question. So once we've expanded to the species level, what you can actually do is jump into this coverage visualization. It's this little, almost looks like a bar graph, that icon. And if you click this, this will actually pop up our coverage viz, where you can start to look into the coverage across the genome. And actually, if you click on this down arrow, you can switch between different accessions as well. We do have the ability for you to go in and look at the read level visualization if that's something that you're interested in, or we have links out to the NCBI entry for this particular accession. And you can start to see, in this case, this is extremely good coverage, but this is where you can find the coverage depth and breadth metrics. And then the second part of that question, does the pipeline do any de novo assembly of metagenomes before sequence ID? So we actually do de novo assembly using spades, and we output the resulting contigs in our report page. It's also possible to download all the reads associated with a particular taxon and reassemble them using offline pipelines specifically tailored to that pathogen, if that's something that you're interested in. Cool, I will hang on here for a minute or two. Oh, can I download the mapped viral sequence? So what you can do is you can download the contigs here. All I did here was I just highlighted over the contig that I'm showing in this visualization, and I can actually just click on that download config FASTA. We do offer a couple other ways of downloading this material and the data that's presented in the report. So if I go to this upper right-hand corner, I'm presented with a couple different options in terms of download options. So if you, right now, I believe if I chose this, let's see, this might just kick off this just kicks off the download right away. So here I'm not able to specify a particular taxon that I want to download, but you could download all of the non-host reads or all of the non-host contigs. Another thing that you can do that we actually recently added to IDseq, if you go back to our project, one of the things you can do now is actually select a multiple samples and kick off a download, like a different type of download across multiple samples. So one of the things that you can do is actually select let's see, where is it? Yeah, so you can just select any of these different download types. Some of them will prompt you for additional metrics that you want to include in your results. And then you can just, once I have filled this out, I can keep, once I press this start generating download, that'll actually kick off the download in the background because some of these are really huge files. They can take a little bit to download, and then you can come back into the tool and find that finished download and start working on the results from there.